Welcome to this explainer on the microscopic hero's journey happening inside you right now. In the exact time it takes us to go through this, your body is going to replicate literally millions of cells. We're diving into the architecture of cellular replication and division, specifically the cell cycle and mitosis. It's this incredibly elegant, highly organized factory process that builds and maintains who we are. So, let's just jump right in.
Think about this for a second. How exactly does one cell become two? Like, how do we grow from a single tiny cell into a whole complex human being? Or, how does your body just know how to heal a paper cut or replace the skin cells we shed every single day? Well, the elegant solution to all of these biological mysteries is the cell cycle. It's absolutely the engine of life. To break this all down, we've got 5 key areas on our roadmap today. 1. The engine of life. 2. Interphase, the active preparation. 3. Mitosis, the great divide. 4. Cytokinesis, the final split. And 5. Checkpoints and quality control. All right, section 1, the engine of life. When you zoom out and look at the macro view, this biological process is actually divided into just 2 primary stages, interphase and the mitotic or M phase. Think of it kind of like a major theatrical production. You've got the long, intense rehearsal and stage prep, right? That's interphase. And then you have the fast-paced, high-stakes main performance, which is the M phase. This isn't just random chaos inside your cells. It's a highly ordered series of events that is absolutely essential for growth, tissue repair, and reproduction in all living organisms. Moving on to section 2, interphase, the active preparation. Now, this is by far the longest phase of the cell cycle. But listen, there's a really common misconception we need to clear up right off the bat. Interphase is definitely not a resting phase. It's incredibly active. Back in the day, early scientists looking through microscopes thought the cell was just napping because it wasn't physically splitting apart. But under the surface, the cell is working overtime, performing metabolic activities and doing all the heavy lifting required to get ready for division. It's for sure not just taking a break. This preparation happens chronologically across 3 distinct substages. First up is gap 1, or the G1 phase. This is where the cell physically grows in size and pumps out essential proteins and organelles. Next, we hit the S phase, or synthesis phase. The stakes here are super high because this is where DNA replication actually occurs. Every single chromosome is duplicated into sister chromatids, which ensures the genetic material is fully copied. Finally, there's gap 2, or G2 phase, where the cell grows a bit more and synthesizes the exact proteins needed to form spindle fibers for the upcoming division. That brings us to section 3, mitosis, the great divide. The prep work is done, and now we enter the intense climax of the cell cycle. Mitosis is where the nucleus of a single parent cell actually divides to produce 2 genetically identical daughter cells. This is a rapid-fire sequence broken down into 5 distinct physical stages: prophase, prometaphase, metaphase, anaphase, and telophase. Let's look at exactly how a cell unzips and separates its most precious cargo, its DNA. Let's take the first 2 steps together. In prophase, the cell essentially packs its bags. That loose, stringy DNA, the chromatin, tightly condenses down into visible chromosomes. It's kind of like taking a tangled ball of yarn and winding it tightly so it's easier to move around. The nuclear membrane starts to break down, and spindle fibers begin forming. Then, in prometaphase, that nuclear envelope completely disappears, and those spindle fibers actually attach themselves to the chromosomes at a specific center point called the centromere or kinetochore. Next is metaphase, and this is where things get really beautifully organized. The chromosomes align perfectly at the equatorial plate. Those spindle fibers essentially tug and pull until all the duplicated chromosomes are lined up dead center in the middle of the cell. This perfect lineup is absolutely critical, guys, because it guarantees that when the cell finally splits, the distribution of chromosomes will be exactly equal. Neither new cell gets shortchanged. And now the action peaks. In anaphase, the sister chromatids get forcefully pulled apart by the spindle fibers. They separate and are dragged toward the opposite poles of the cell, becoming fully independent chromosomes. Then the dust settles in telophase. The chromosomes reach their opposite sides, 2 brand new nuclear membranes begin to reform around each set of DNA, and those tightly packed chromosomes decondense back into that relaxed chromatin state. So now the cell has 2 complete, identical nuclei. Which takes us to section 4, cytokinesis, the final split. So we have 2 nuclei, right? But technically, it's still one giant cell. Cytokinesis is the final physical division of the cell's cytoplasm, resulting in two distinct daughter cells. But the way this happens is actually totally different depending on the cell type. In animal cells, which are relatively soft, a cleavage furrow forms. The cell literally pinches inwards around the middle until it snaps into two. But plant cells have rigid cell walls, they can't just pinch. Instead, a cell plate develops straight down the middle, basically building a brand new wall from the inside out to separate the two new cells.
Finally, we have Section 5, checkpoints and quality control. This whole process is incredibly complex, right? And there are so many ways things could go wrong. To prevent any biological disasters, the cell relies on a super strict internal quality control system run by regulatory proteins, specifically cyclins and CDKs. They run 3 major checkpoints. At the G1 checkpoint, the cell checks its size and looks for DNA damage. At the G2 checkpoint, it verifies that DNA replication went perfectly. And at the M checkpoint, it pauses to ensure all spindle fibers are properly attached before separating the chromosomes.
But what happens when this perfect system breaks down? Well, if those regulatory checkpoint genes, like tumor suppressors or oncogenes, mutate, the cell might just blow right past those stops. It starts dividing uncontrollably. And this uncontrolled cell division is exactly what cancer is. It's the direct result of the cell cycle's quality control failing, which leads to abnormal cell growth and tumors. That's why understanding this cycle is quite literally the bedrock of modern cancer research. Let's zoom back out for a second and look at the macro impact of all this. Every time a child grows taller or you heals from a scraped knee, that's mitosis at work. It repairs damaged tissues, replaces dead cells, enables asexual reproduction in some organisms, and most importantly, it maintains our genetic stability. This continuous microscopic replication is what keeps all eukaryotic organisms alive and perfectly balanced. So I want to leave you with this thought. What would happen to our bodies if this perfect microscopic clockwork just stopped, even for one single day? The fragile balance keeping us healthy, growing, and alive right now is all managed by this brilliant architecture. Thank you so much for tuning in to this explainer. Keep questioning, keep learning, and I'll catch you in the next one.